So with that, I'm just going to hand it over to Hongxi, our own Hongxi, to introduce our speaker today. And thank you for coming, and please continue. So, Hongxi.
Yeah. Hello, everyone. Dr. Santiago Coelho was my previous colleague at NYU. So he's a postdoc fellow in the radiology department at NYU School of Medicine, working with Professors Dmitry Novikov and Errol Spearmans. His work spans MRI biophysics, tissue modeling, estimation, optimal experimental design, and accelerated microstructure imaging. And Dr. Coelho is an NIH K99 awardee and an ISMR Junior Fellow. He's an excellent MR physicist and also a very good, talented soccer player. Welcome.
Thank you, Hongxi, for the very kind introduction. Let me put the timer. Good. And I'm very sorry for those of you who cannot eat pizza today. I hope that you still stay for the seminar. And yeah, I wanted to also thank Hongxi and Susie for having me here and you for attending. So let's start first with a bit about me. I'm originally from Argentina. I come from a small city near Buenos Aires, maybe 4 hours by car from Buenos Aires, where I did my undergrad in EEE. Then in 2015, I moved to the UK to do a PhD in biomedical engineering. And then in search for better weather in 2020, I moved to NYU, where I met Hongxi. And since then, I've been working on MRI biophysics with Al Spearmans and Dmitry Novikov. I like teaching quite a bit. I've done it quite a bit across all these countries, and I'm the stereotype of an Argentinian, so I really like barbecuing, and as you said, I really like soccer. But that's enough about me. Let's talk about scales, right? This is a biomedicine talk. And as you know, there's a ton of scales, a wide range of scales involved in our typical day-to-day applications. But in this talk, we're gonna first start with these two, right? On the one hand, we have the molecular scale on the order of the nanometer level. And here's where our NMR signals originate, right? On the other hand of the spectrum, we have the microscopic scale, which is commensurate with our image resolution, right? Typical MRI images and images from other imaging modalities like ultrasound, PET, CT, they live in this space over here, right? Which is essentially millimeters, 10 to the minus 3 meters. But this talk is going to focus on this scale that is somewhere in between of these two, which we like to call the microscopic scale in the order of the micrometer. And the reason why I'm interested in this scale is because pathological processes originate here. And what I hope to convince you after this talk is that we can probe this scale of cells in vivo, and we can hopefully use this information to develop early disease biomarkers. I don't need to tell to this crowd the good things about MRI, but to sum it up, it's non-invasive imaging modality. It can image whole organs. It provides us with multiple contrast mechanisms and can probe structure and function. So it's amazing. But why then do we want to do diffusion on top of it? Well, if you look at conventional 3D structural MRI scan, where resolution is in the order of the millimeter, and you look at what tissue looks like at the millimeter scale, you can see there's a ton of heterogeneity there. We need to zoom in significantly in order to start disentangling different types of cells, right? And if you want to actually get voxels in this scale that I was telling you before that we're interested in, we actually, if we want voxels in the order of microns, we actually need like a boost in resolution in the order of 100 or 1000, right? And the problem with this is that MRI resolution is SNR limited. So it's tricky to actually build a better or a larger scanner to actually achieve this, because essentially if we want to keep the same SNR and we want to, as we have in say 1 millimeter cube voxels in 3D, we actually gonna need a main field in the order of the thousands of Teslas, right? So we cannot really buy ourselves a bigger scanner to solve this resolution problem this way. An alternative is to use the diffusion of water molecules. And during our experiment, water molecules are randomly moving, right? And in a free medium, their mean square displacement due to diffusion is exactly proportional to time, right? Where this proportionality coefficient is nothing else than the diffusion coefficient of the medium. The nice thing about this proportionality is it allows us to convert time into distance, right? So if we plug in experimental times that we have in MRI together with the water diffusivity coefficient at body temperature, what we get is that the typical experimental lengths that we're sensitive to during our MRI experiment are in the order of 1 to 50 micrometers, right? And this is what gives us the microscopic scale sensitivity that I told you at the beginning I was interested in. So this kind of means that we could use, in principle, diffusion MRIs and in vivo microscope. And the idea of my research goal is to try to develop fast, physics-informed ways to reduce the dimensionality of MRI signals and to use this to create meaningful biomarkers, microstructure biomarkers. And as a disclaimer, a lot of this is a neuroimaging talk, but a lot of these things have actually been applied and we are trying to apply outside the brain. So a brief outline of the talk, I'm gonna continue a bit with why do we want to use diffusion MRI and what do we measure. Then I'm gonna show you a bit of my work on microstructural fingerprints and go from diffusion tensor imaging to higher order terms in the cumulative expansion. Then I'm gonna switch gears a bit and show you a bit of our work on biophysical tissue modeling for the brain, both white and gray matter. And then I wanted to give you a bit of a short discussion on what are my plans for my future research. Please ask questions. Interruptions are welcome at any time during this talk, so don't be shy. So let's go from random walks to diffusion signals. In diffusion MRI, our signal is just the average phase of all spins that we have in a voxel, right? And this phase is going to be given by the integral of the diffusion encoding that we apply times the random displacement of each spin that each spin has, right? And the simplest way in we can measure this is to use what we call the pulse gradient spin echo diffusion encoding, right? Here we just use magnetic field gradients, which we apply in a given direction, and we essentially apply two pulses. The first pulse to label spins positions, we wait for a given time, and then we apply the opposite pulse, opposite polarity, to unlabel them, right? And what happens is that essentially, if spins move along the direction G between these two pulses, the signal will decay, right? It's as simple as that. And this means that the signal that we get with diffusion is a function of these three experimental parameters, right? Of the pulse width, the pulse amplitude, and the separation of the pulses, right? As I was saying, in a simple case of diffusion, which is what we call Gaussian diffusion, where mean square displacement is proportional to time, essentially all this dependence on these three parameters gets collapsed to a single parameter, right? And this was what the scalar parameter that's typically referred in the diffusion literature as the b-value, right? So essentially it gets simplified, just depends on a single acquisition parameter. But in reality, there's nothing stopping us for going beyond this type of diffusion encodings, right? The experimental space of diffusion is much richer than this. We could play arbitrary waveforms, encoding waveforms that vary as a function of time, and we could still get diffusion measurements, right? So in reality, it looks like this. Our signal is a function of the waveform that we play. Again, if we simplify our case of study to environments that exhibit Gaussian diffusion, then all this complicated dependence gets simplified, that the signal depends, again, not on a scalar b-value, but on a second order tensor called the b tensor, right? That essentially generalizes this dependence on b. But again, it's much simpler than just functional. And so just to highlight, anytime throughout the talk that I'm going to be talking about b tensor, I'm somehow implying a Gaussian distribution of phases here, at least at the compartment level. Otherwise, one would need to specify the full waveform in order to make this work, in order to use these free waveforms. So let's start on the bit of microstructure fingerprints, and let's start with DTI, diffusion tensor imaging. As I told you, the simplest case of diffusion is what we call Gaussian diffusion, and the simplest case of Gaussian diffusion is isotropic diffusion, right? And in this case, the proportionality between mean square displacement and time is the same, independent of the direction, right? So what we can do is we can take a bunch of measurements and for different experimental parameters p, and when we plot the log signal, we'll see that this shows a linear decay, right? So the signal as a function of e is a monoexponential. And the slope of this measurement measurements in the log scale is nothing else than the diffusion coefficient. The next step, sort of in complexity, is to say, okay, diffusion is still Gaussian, but it's anisotropic, right? Depending on the direction in which we are looking at, molecules can actually spread faster. And essentially, if we take measurements, again, for different b's, it will also look monoexponential. It's just that the slope, depending on the direction, will change. And the way to mathematically represent this is, instead of with a scalar diffusion coefficient, it's with a second order diffusion tensor, right? And this is kind of the basis of diffusion tensor imaging. So as I was telling you, in this case of where we have Gaussian compartments and we have b tensor encoding, our signal is, again, simple, a monoexponential, where we have a second order tensor that reflects the measurement, b tensor, and a second order tensor that reflects the tissue, the diffusion tensor. For the diffusion tensor, we can, for each voxel in the brain, we can estimate it from measurements, right? And we can plot it like as an ellipse, right? Highlighting the main direction of what the diffusion, the second direction, and the third. And then we can compute this throughout the brain and color code them depending on which direction they point, right? Then we can draw streamlines that connect that travel as the direction of the main eigenvector of this tensor, and then draw these beautiful trajectories. On the measurement point of view, this bit tensor, as I told you, is nothing else than the integral of the gradient waveform, right? And it's a symmetric tensor, right? So it's like out of this 3x3 matrix, there's only 6 independent parameters. And what's interesting is that throughout the diffusion literature, the vast majority of papers focus on this specific type of tensor, which is sometimes referred as linear tensor encoding, which is typically what you get with the post-gradient spin echo sequence. And what this means is essentially that when you apply diffusion encoding in this way, you're sensitizing your signal to displacement along a given direction, right? And that's it. And the way to see this in the B tensor is that the B tensor is rank 1. So it has one non-zero eigenvalue and 2 eigenvalues that are 0. But in principle, nothing's stopping us from generating B tensors that have arbitrary shapes, right? In this case, this is planar encoding. It means that we're sensitizing for diffusion along a plane with 2 equal eigenvalues. So it's essentially any displacement that can be projected on that plane will produce signal alternation. And that gives us a functionally different signal dependence on experimental parameters. We can also do spherical encoding. Essentially, it doesn't matter the direction. As long as molecules diffuse, that will create signal alternation. Or we can go somewhere in the middle where we have arbitrary asymmetry of the B tensor. So all of these are options that are possible. And then, of course, the size of this B tensor, which is the trace, is what we are used to referring as the conventional B value, right? So it's the sum of the eigenvalues. In this case, it's 1 eigenvalue plus 0 plus 0. So it's really the same thing. So if you have looked at diffusion data, you can probably see that if you keep increasing the experimental parameter, this B value, and you plot, again, measurements for real tissue, you'll see that at higher diffusion weightings, signals start to deviate from this linear decay that we typically see in DTI data. And this is because we become sensitive to the non-Gaussian contribution of the diffusion process, right? And the sources can be multiple. For example, it can be due to having multiple heterogeneous compartments. It could be because there's water exchange between the compartments. It could be because there's bound walls causing restrictions to the diffusion process. It can be many of them. But the simplest way to represent this mathematically is to say, okay, yeah, let's take the log signal. It's not linear, so let's add a quadratic term. And this quadratic term is what is referred to as the kurtosis part of the signal, right? In this case of isotropic media, it's very simple. It's scalar on B term and scalar on B squared term. But if we want to do an isotropic media, then we just need to make sure that this term that we are on the fourth order is actually a fourth order tensor. And this is what's essentially the basis of diffusion kurtosis imaging or DKI. And this kurtosis tensor, just like the diffusion tensor is fully symmetric, this kurtosis tensor is fully symmetric, and it has 15 independent components in 3D. This expression over here may look a bit scary, but it's what we call the cumulative expansion, but it's actually not at all. It's just taking the log of the signal and expanding in powers of B. This is just a B part, B squared, and we could keep going. However, there's some issues with conversion, so that's why typically we go up to B squared and not anymore. But so far I showed you that diffusion in the brain is anisotropic, right? And that means that when we feed diffusion tensors, these diffusion tensors are anisotropic. And this means that, for example, if we look at this voxel, which is representative of this diffusion tensor, right? Most axons are aligned this way. We measure this direction. We're going to measure small signal because there's fast diffusion, so there's a ton of attenuation. If we measure this direction, we're going to measure more signal because there's slower diffusion. But if we rotate the voxel, suddenly the meaning behind these two measurements will change, right? So if we're after information about the tissue microstructure, this information cannot be dependent on the local orientation of the voxel with respect to scanner, right? It needs to be independent of this. And what it means is that we are essentially after rotational invariant information from the diffusion measurements. The simplest way to compute rotation invariant information is the following. Say you have the diffusion tensor, right? I told you it's symmetric, so 3x3 matrix, it's only 6 degrees of freedom. Since it's a tensor that lives in 3D, we know that we need 3 angles to specify its orientation, right? The 3 Euler angles. And once we have this, essentially the remaining degrees of freedom should be mapped. We should be able to map them to rotation invariance. And that's actually, yeah, 6 minus 3 is 3. That's the 3 eigenvalues of this matrix over here, right? Those are the 3 invariants. There's nothing more to it. With these 3 eigenvalues, these invariants, you can compute all the DTI maps that are rotation invariants. For example, mean, axial, radial diffusivity, or fractional anisotropy. All of these are functions of this, right? So the idea of this first part of the talk is to show you how can we generalize this concept of extracting rotation invariants from the diffusion tensor, but to acquisitions that contain arbitrary diffusion waveforms, and to do it for higher order cumulant tensors, like the kurtosis tensor and tensor and the covariance tensor, which I'm going to show now. So within the long diffusion time regime, essentially, we can make a sort of quite a general picture of what voxels from tissue look like. And let me walk you slowly. Essentially, we have an MRI voxel, which is essentially the scale is roughly in the order of the millimeters. That's the order of magnitude. But as I told you, the diffusion length is between 1 and 50 micrometers, say around 10 micrometers. That means that to our signal, we have a lot of water pools that are contributing to it, right? And in principle, these water pools may not be identical, right? There may be some heterogeneity for each of them. And also they may be anisotropic. So kind of the most general picture within this framework is to say, you know what? Each of these water pools is represented by its own diffusion tensor. And therefore, the most general way in which we can refer to a voxel is to say a voxel is a collection of these diffusion tensors. It's an arbitrary distribution of these diffusion tensors. The signal from such a collection is nothing else than signal from a single compartment integrated over the whole distribution, right? And this is what angular brackets denote here. But what is this arbitrary concept of the probability distribution of diffusion tensors in voxel? Well, it's nothing else than the voxel microstructure. One can think of it as ad hoc distribution families, like Wishart distribution or gamma distribution, but we can also think about microstructure-inspired distributions, like, for example, the ball and stick model. That's a distribution of diffusion tensors. The standard model, that's another one. NODI, multi-tissue constraint spherical deconvolution, and so on. There's actually, this is a wide range of models fall into this framework. So we can take the log of this expression and then Taylor expand. And if we write the first two terms, we have here the fourth order cumulant expansion. Note that these are voxel level tensors, right? So these are tensors that somehow summarize information about P of D. And the nice thing is that they make no assumptions. So this probability distribution of D can be whatever it wants, say, but when we do this estimation, we are not really kind of constraining it to anything. We're just getting sort of, you could argue, different moments of the distribution. And actually, if you look at the meaning behind these two tensors, the meaning behind them is quite intuitive. The first term, which is the diffusion tensor, so the B term in the cumulant expansion, is the mean of the distribution. And that's kind of completely reasonable, right? If we have a distribution of diffusion tensors in a voxel, and we do a DTI estimation, we're actually being sensitive to the mean over that distribution. On the higher order term, the covariance tensor, we're essentially capturing the covariance between these tensors, essentially how heterogeneous they are. And note that this covariance tensor I'm writing here, you could think of it as a sort of generalized kurtosis tensor because it has fewer symmetries. But the point is that kurtosis is part of it, and it has also some other stuff apart from kurtosis. And what I want to show you now is that we can use representation theory to essentially classify all these 21 degrees of freedom that I'm showing you from these covariance tensors, just like we did for the diffusion tensor before. And yes, since it's also a tensor that lives in 3D, 21 minus 3, 18, we essentially should be able to define 18 different rotation invariants. To start simple, let's focus on the diffusion tensor, right? We know diffusion tensor is a second order tensor. We can write an irreducible decomposition of it, right? Splitting the isotropic contribution and the anisotropic contribution. And you can see that the parameter count holds really well. You have 6 degrees of freedom here. 1 comes from the isotropic part and 5 comes from the anisotropic part. This is very similar to what to those of you who have worked with spherical harmonics, to a spherical harmonic decomposition, right? We can write any ellipsoid as a sum of 0th order and 2nd order spherical harmonics. But for the covariance tensor, we have a product, right? And this is where things get a bit nasty because we have D times D. And the thing is that when we have, this is a sum of irreducible representations, and then we have a product. When we have a product of irreducible representations, the product actually is reducible. So we'll see that newer sort of elements start to show up. And the best way to show this is to write this 2x2 matrix where we can see cleanly that we have all the products, right? We have when D0 is times D0, so essentially it's isotropic part times isotropic part. That part of the covariance tensor is essentially giving us information about the size variance. So essentially, how heterogeneous are these compartments in the voxel in terms of their size, in terms of the individual mean diffusivities. Then the next term is, say, when it's D0 times D2, right? And these terms are the size shape covariance, right? It's essentially how do these compartments covary with size and shape? Are the most anisotropic ones the smallest ones, or the most anisotropic ones the larger ones, right? This is what this second order tensor contains. And then here, when we have D2 times D2, that's actually where things get messy, because even though it's a product of a single representation 2 times, it splits into a sum of 3 representations: first order, second order, and fourth order. And all of them contain information essentially about the shape covariance. How do the shapes of the compartmental diffusion tensors vary together? So you can write this irreducible decomposition where we have 2 isotropic parts, 2 anisotropic parts from second order, and 1 anisotropic part from fourth order. And the parameter count holds nicely because it's 21, it's 1 plus 5 plus 1 plus 5 plus 9. And if you are interested in the math behind it, you can check this paper here. But essentially, this decomposition is valid for any fourth order tensor with minor major symmetries. It doesn't have to be diffusion. Yeah.
Sorry, so for this fourth order approximation, are you keeping terms that are 0 times 4?
So in principle, from this part, we have 0 times 0, 0 times 2, and 2 times 2. So the 4 comes from splitting 2 times 2, because it's like that follows the addition of angular momenta. So you're adding up 2 and 2, then you have as a result 0, 1, 2, 3, and 4. But the 1 and 3, they get cancelled because of this has minor major symmetries, so that kind of kills the odd terms.
Right.
So the 4 comes from 2 times 2.
Okay, so we're not keeping anything from 0 times maybe another term within the D.
If you wanted 4 from 0 times 4, you would need to go 1 extra order to
Right.
DDD. So it would be the next one after the covariance tensor.
Right.
So the V cube term. There you would have it because you have D0 plus D2 times D0 plus D2. That gives you a 4 somewhere there times D0 plus D2. So then you would have 0 times 4.
It will make it very complicated. I probably don't want to do it. It's just a makes me think that the fourth order that we have here is some part of the fourth.
What do you mean the total fourth order?
So there are terms coming from 0 times 4 that
So let me put it this way. There's only one source of fourth order at the B squared. The fourth order part of the kurtosis tensor is the same as the covariance because there's only one, and it comes from doing D2 times D2. That's kind of like there's no other source, because then you have 0 times 0, that gives you 0, 0 times 2, that gives you 2, and that's it.
Right. There's no other source being ignore
Higher order terms. Yes, exactly. Yes, yes, exactly. But that was a really good point. And yeah, this can be applied, for example, to elasticity tensors. So that's another thing about this irreducible decomposition is quite general. But then, if we can get all these different independent blocks, we can actually then try to compute rotation invariance for each of them, right? And that's what I'm going to show you next. But to sum it up, using representation theory, we can, from complex objects, build much simpler ones that are irreducible components, right? And the nice thing about these components is that they transform separately under rotations, and this kind of helps us to study them separately. And of course, we can compute rotation invariance from each. And these are some examples of all the maps that we can get from clinical scanner. This was like under less than a 10-minute protocol. But the whole point of this is to show that only using a few of them, actually 4 out of these 18, plus the invariants from the diffusion tensor, which are really well known, with those 7 invariants, you can actually generate all the diffusion contrasts that you have seen, like mean axial radial kurtosis fractional anisotropy, microscopic fractional anisotropy, isotropic and anisotropic variances. So the rest kind of have been really underexplored. And just to show you some preliminary results I have is we have a huge 2-shell data at NYU, right? where a lot of people have tried to do DTI and DKI. And if you try, that dataset has a lot of control subjects and a lot of MS subjects. If you try to estimate a logistic regression that tries to do the solve the classification problem of detecting controls from MS, you try to do that with DTI rotational invariance, you get some result. Of course, you use the two shells of the protocol, and then you try to do it with DKI invariance, that result improves. But the nice thing is that without having to redo the fit, just taking the same kurtosis tensor, but squeezing some more rotation invariants that are, again, as I showed earlier, that come from different physical origins and therefore are mathematically independent, you can actually see that the classifier improves the performance. So this is kind of hinting that the moment you add more invariants, again, that they need to be independent, they cannot be a combination of the ones you already have. It leads to a better discrimination. And that's kind of a nice empirical result that can kind of complements the theory part. So to summarize this bit, we can get, once we get to intermediate diffusion weightings, so that we see some curvature in the signals, we can fit the cumulative expansion and apply our favorite irreducible decomposition. And from it, we can just compute all these family of invariants. And then we can, for example, build machine learning classifiers that try to exploit all these fingerprints. And this is what I was referring earlier as microstructure fingerprints. It's kind of tricky to add meaning to them, but the nice thing is that they really contain complementary information. And with the meaning part, I'm gonna come back later in the second part of the talk. And yes, I quickly wanted to show you a bit about fast acquisitions in the sense that I showed you that this covariance tensor splits into different irreducible components. And maybe I think an interesting question to ask is, what if we're not interested in all the parts? What if we just care about some bits of these higher order tensors? Do we really need to measure the whole thing, compute then the one we want, and discard the rest? Can't we just, in the spirit of compressed sensing, can we just try to measure less and acquire only what we need? And the idea is that yes, we can. And in this case, with this irreducible decomposition, we can actually see that if we use spherical designs, so essentially special sets of directions in 3D that cancel higher order anisotropy and just keep the isotropic components, we can quickly just grab these two components and cancel these other ones. And therefore, we end up measuring them with much fewer directions or measurements than the ones we would need to measure the whole thing. So with this, like, for example, we can measure from, in theory, with 7, but for robustness, we use 12 diffusion-weighted images, and we can get mean diffusivity, mean kurtosis, fractional anisotropy. And this can be done in a Prisma in roughly 1 minute for 2-millimeter isotropic resolution. So it can be done really fast if you neglect all these higher order terms if you cancel contributions from these higher order terms that you don't want. The same if you add some spherical encoding data, in theory 8, but for robustness, you can use 15. And this gives you like a 2-minute protocol that can add microscopic fractional anisotropy to the mix. And I think this is kind of a nice sort of sub-result of this project, that it's nice to understand all these different blocks of information that come from these higher tensors, but that allows you to find some sort of tricks to try to do experimental designs that focus on certain bits and actually become much, much cheaper in terms of scan time. And just to show this is quite recent. This is a project I collaborated with Hongxi, where essentially we're trying to develop machine learning estimators that account for gradient nonlinearities. So nothing to do with what I just talked to you about. But the thing is that, as a side note of that project, we also use the irreducible decompositions, not for the tissue in this part, but we used it for the diffusion encoding. Essentially, when you have gradient nonlinearities, the diffusion encoding that you end up applying deviates as you move away from the isocenter, and this is kind of a map of how the p-value and the direction of the v-vector get deformed as you move throughout the field of view. This is for a Magnus gradient insert. But it's nice that if you take this gradient nonlinearity tensor that is essentially driving telling you you need to adjust these diffusion weightings, you can actually get the isotropic part and see how that changes spatially. And then you can also separate its anisotropic part, right? So essentially this map is showing purely how the overall p-value gets increased as you move away from the isocenter, while this is showing you the standard deviation of how, as you change directions, the p-value changes, right? So the mean of this guy here is 0. It's just telling you that there's some deviation purely depending on the directions. So this is a nice way to separate sort of isotropic versus anisotropic contributions. And yeah, this is not really about tissue. This is about the scanner itself. So I think that's kind of a powerful message about the reducible compositions. To sum up this bit, let me tell you that these fingerprints, of course, they add great sensitivity to microstructure. As I showed you, these rotation invariants of the cumulative expansion add complementary tissue properties to DKI and DTI, and you can see that when you try to do like logistic regression, for example, which is a very simple approach, but it's already enough to show it. But of course, these outputs, these invariants are really not so specific. If we want specificity, we're going to need biophysical models, and we're going to need to focus on each tissue separately. And that's what I'm going to discuss a bit now. And I'm going to start with the white matter. So, as I told you before, the diffusion length is what defines local compartments. And if we focus on the white matter, we could say that these local compartments contain essentially a local fiber bundle, right? And that bundle can contain axons, extracellular space, and so on. So say this is an EM image of a white matter voxel. You can see that there's a lot of white matter of axon bundles, right? So in the diffusion length that we have, our compartment essentially consists of a lot of axons, the space around them, and so on. And the idea is that in this bundle, we essentially contain all the microstructural information that we want. Essentially, it contains the diffusion and relaxometry and water fraction properties of the axons, of the space surrounding the axons, of free water if there is some, and everything. The idea, also, the point is that within a voxel, as it's shown in this image, there's more than one bundle, right? So essentially, if we want to compute the signal from such an arrangement, we just need to add the signals from each of the bundles. But the signal depends on the bundle orientation, right? Because it's diffusion, and if the bundles are anisotropic, so essentially we need to use this object, which is essentially a fiber orientation distribution function, which is what tells us how these bundles are aligned within the voxel, right? It's nothing else than a probability distribution of the sphere that tells us exactly how many bundles we have pointing in which direction. So with all these ingredients, essentially we can say that our diffusion signal is a convolution of the response function of a given bundle times the orientations that they have, right? The fiber of the F. And that's why it's convolution. K here is kernel, which is the response function, and P is the F of the F. And if you've looked at the white matter literature of diffusion models, you'll see that there's a ton of papers that they look very dissimilar. The same if you look at gray matter modeling, and the same if you would look at axon diameter mapping. And this may look like a zoo of models out there, but in reality, all these papers are just the same thing, in the sense that the meta concept lying around there is that they all rely on this spherical convolution assumption. They're all spherical convolution models. The only difference is that some of them apply certain constraints on the F of the F, right? Some of them allow it to be an arbitrary expansion of spherical harmonics. Some others say parallel fibers, crossing fibers, whatever. And some others apply several constraints on the response function, say only one compartment, only two compartments. Compartments need to have fixed diffusivity values, and so on. But essentially, all these models have more in common than not. In the case of the white matter, if you try to say, okay, let's be as relaxed as possible with these constraints, what can we come up with? That's what many people have called the standard model for white matter. It's kind of a generalization of, like, you could say, no, the fiber body imaging, standard model, spherical mean technique, WMTI. And the idea of this model is saying, okay, we have intractional signal, we have extractional signal, we have free water. They're not in exchange because axons are myelinated, so they're impermeable. And all of these compartments, each with their own diffusivity and, if you want, relaxometry properties, they define the standard model bundle or fiber response or kernel, as you want to call it. And essentially, our signal in white matter is the convolution of that fiber response together with an FODF. And the FODF can have, again, as many peaks as you want. It can be general. The nice thing about this model is that it gives us different microstructural information specific to different pathological processes, depending on where you look at. In the case of the microstructural parameters, so the parameters of the fiber bundle, you have axonal water fraction, axonal diffusivities, extraxonal diffusivities, free water fraction, and all of these have been linked to different pathological processes. On the case of the fiber ODF, you could, for example, expand it in spherical harmonics, like it's done most popularly by constrained spherical deconvolution approaches, and you can get your fiber ODF. And with the fiber ODF, you can do tractography. You can do presurgical planning with that then. So it has a lot of applications depending on whether you look at information coming from the bundle or the kernel, or information coming from the FODF. But not everything is so nice, unlikely. And since this model is a really nonlinear model, depending on the dependence of the parameters on the measurements, it's a really hard inverse problem. And this means that typically parameter estimation from conventional linear encoding protocols is degenerate. It's like trying to distinguish these two shapes using only this projection. They will look identical, and it's going to be hard to say which one is which. So what we need is essentially to include orthogonal measurements so that we can actually separate which shapes are we talking about. In the case of diffusion, this type of orthogonal measurements can be, for example, double diffusion encoding or B-tensors that are not like these sticks that have only one non-zero eigenvalue, but have other shapes, right? Non-linear encoding. And this is some of the work I did during my PhD, where essentially we showed that with linear encoding, in this case of the general version of the standard model, you have an infinite number of solutions with even 2 branches, so it's like crazy stuff. But the moment you add 2 different types of tensors with different p-values, you can guarantee that the solution is going to be unique throughout the combination of ground truth values. And that's kind of nice. But this begs the following question. We want different p-values in our acquisition. And I just told you that to guarantee uniqueness of solution, we need different shapes of this p-tensor. So even if we focus in these actual symmetric B-tensors, the question is, what should I pick? And this is some of the things I did during my postdoc at the beginning, which is essentially saying, can we optimize our acquisition so that, say, we have certain constraints. We have an acquisition time that we cannot exceed. We have certain gradient Howard limitations. And we want our data, for example, to be compatible with big consortia studies like HCP or UK Biobank. So we need, say, those typical 2 shells, B1000, B2000. And the nice thing is that so essentially what we did is we derived the cost function that essentially couples our parameter estimation together with our with the experimental settings, and it nicely predicts the root mean square error of your estimates. So by minimizing that, you're essentially picking the measurements that are the most informative for your problem, for your estimation. And what we saw was that apart from the conventional 2-shell protocols, there's 3 regions of the experimental diffusion space that keep coming up. And the first one is just go to high b-value linear tensor encoding. The second one is intermediate to high b-value anisotropic b-tensor encoding, but not as anisotropic as linear. And then the third one is spherical tensor encoding in the intermediate diffusion weighting range. And this happens to be super consistent across scanners because, of course, when one does this optimization, we need to take into account that for certain hardware, it's more expensive in terms of echo time to reach higher v values. So you need to account for all these trade-offs that it's not completely for free to take these different measurements. And of course, the next question that comes after doing this is, okay, what if I also want to vary echo time, right? Because if I have all these compartments I told you about, they are very likely to have different relaxation properties. So if they have different relaxation properties, if I vary echo time, I'm going to attenuate them differently at different echo times. This should help with the inversion so that I can separate them further. And that's something you can do, and it actually works really well. You can design a diffusion relaxometry protocol where you vary b-value, b-tensor shape, and echo times. And this gives us pretty good results. You can check the estimator here and the paper here. But essentially, you can get pretty good scan-rescan coefficient of variation, both at the voxel level and at the ROI level. And the thing is that that's still this protocol here took around, say, 25 minutes. So later I'm going to discuss how can we do to speed this up. But as I told you earlier, this convolution framework is actually really powerful. So even staying within this convolution, we can go beyond the standard model, right? So there's nothing stopping us to add to modify this fiber bundle functional form and to add other stuff. For example, ways to go beyond the standard model would be change the compartments and make them non-Gaussian. Why would we want to do that? For example, if we want to do finite axon radius, so axon diameter mapping, if we go to really high B, we then become sensitive to displacements perpendicular to the axons, and therefore one could measure axon diameter. And all of this, like connecting, like if one uses the connecting to scanner or the magnet scanner, and all of this would not still violate the spherical convolution assumption. All of this can be done within that framework. You could also focus on water exchange. I know Hongxi has a lot of work on this. And if you go to a gray matter, for example, you could do NEXI or SMEX. Again, all of these are spherical convolution models. Also, you could, if you're interested in characterizing soma, like using Sandy, again, there's nothing stopping you from adding additional compartments to the fiber bundle. This is all within this spherical convolution framework. In case of a gray matter, as I was saying, since the dendrites are not myelinated, they may not be impermeable, and that's why it's of interest to try to measure, to account for exchange in the modeling. And actually, measuring may be useful because characterizing membrane permeability may offer insights into physiological processes. And this is some collaboration we did with Hongxi and Kwok and Susie here at MGH, where essentially they use the Connecting 2.0 scanner to measure exchange times, which is essentially the inverse of the exchange rate, which is related to membrane permeability to the surface to volume ratio. So it's kind of nice that one can really consistently get a measurement of this. The downside is that still we need high grains to do this reliably and reproducibly. So I think that that's also an interesting avenue for future research. And to summarize this bit, I told you that models provide specificity to microstructure, and that standard model parameters in the white matter actually produce highly reproducible values. But as I told you, these acquisitions can quickly become really long, right? Because the moment you change diffusion weightings, you measure a new shell, you add a ton of directions, your total number of measurements goes up. So I'm going to show you now a bit of my work on what I like to call Q-space undersampling, and also some future directions for doing Q and K-space undersampling. I told you that within this spherical convolution framework, we can write the signals as convolution of the sphere of fiber response and FODF, right? And I hope you believe me that this is a really wide class of models that applies to both white and gray matter. We don't like convolutions, they look scary. That's why it's usually nice to go to Fourier basis, right? In Fourier basis, convolutions become products. And in the sphere, that Fourier basis is spherical harmonics. So we can write the spherical harmonics of the signal as a product of spherical harmonics of the kernel times spherical harmonics of the fiber ODF. The problem is that these spherical harmonic coefficients will change depending if the box is oriented one way or the other. That's why it's typically useful to compact them, like taking the L2 norm, and compute rotational invariance of these spherical harmonics, so that essentially we purely focus on the dependence on the microstructure parameters of the kernel. And how does this relate to measurements? Well, you can say I acquire a shell of n uniformly distributed directions. You can linearly estimate the spherical harmonics, and then you take the L2 norms, right? So you end up with rotational invariance of the signals that essentially have information about the kernel. What I want to tell you now is that the microstructural information that you have on your kernel parameters, it doesn't matter what's the functional form of the kernel, but that information is determined by the number of independent rotational invariants that you can generate, right? So it can be tricky, but say you have 30 directions on the sphere. Here I'm plotting way more for visualization. And the anisotropy that you can observe is of degree 4 on the sphere. So essentially, if you do a spherical harmonic fit, you need to account for a total of 15 components, ranging from 0, 0 to 2 minus 2 to 2, 4 minus 4, 4, 4. So these are 15 in total. So what one could naively say, okay, if I have sensitivity to 15 spherical harmonic components, I can fit, assuming my scenario is really good, a kernel that has 15 parameters. Well, that's not correct, because if you actually can compress them to 3 different rotational invariants, then you're not actually accessing 15 microstructural components. You're only accessing 3. And it works like this. The moment you measure a shell, just say you get these 3 invariants. You measure another shell here, you get 3 more invariants. You measure a few more, and then you can essentially, these invariants here, with the process of parameter estimation, you can convert them into the actual model parameters of your interest. Again, this is independent of the model. The constraint that we have here is that the number of crosses that we have here, so the number of invariants that we have in our protocol that we're sensitive to, needs always to be larger than the number of parameters, right? But the problem is, if we're interested in the number of rotational invariants, which gives us sensitivity, why are we acquiring data like this? So essentially, if we explore this very sparsely, the diffusion weighting space, and we keep sampling the directionality every time we change it. So that's kind of a bit inefficient, and the goal of this part of the talk is to show you how we can speed this up. And the way to do this is actually to drop the shells. And the way to do this is to exploit that the fiber ODF is something that is constant. Within this convolution framework that I hope I convinced you so far by so much repetition that it's kind of quite wide and applicable, if you see that, you can see that the signal is convolution of the fiber response, and this is what varies with the v-values and echo times and so on, but the fiber ODF, it always stays constant. So this is kind of already highlighting to us that when we change the diffusion encoding, the source of anisotropy is always the same. So there's some redundancy there that we can exploit. And what we propose to do is something that looks like this. We call it Q-space undersampling. But essentially, you still measure a bunch of directions over the sphere, but each time you change direction, you also change the diffusion weightings. So essentially, if you look at a given p-value, that p-value only has one direction. It doesn't have more. But then you want to look at the other direction, it matches to another p-value. And the idea is that using this spherical convolution sort of assumption and thinking that that's the glue that's tying together all the measurements, you can actually reconstruct all these rotational invariants, but from single directions, right? Without even having to know anything about the kernel. And let me quickly show you how this works in the results. We acquired this protocol on a healthy volunteer in a Prisma scanner. Essentially, we took all these directions on the sphere that do not coincide. They are all different. There are 515 total. And this is a proof of concept. It's not that the method only works with this number. But we acquired 550 directions. And for each of them, we acquire a different combination of b-value and diffusion time. So this is the triangular grid we acquired. And the colors match, like this red here should be somewhere here, this blue here would be somewhere here, and so on. And the idea is that we also, for these black crosses over here, acquired validation shells. So that in here, I acquired 60 directions at that specific value and diffusion times, so that I can compute rotation invariance from there and compare to the rotation invariance computed where I only have one direction. And yes, as I said, this is a proof of concept, but just to show that one way of computing the invariance would take us a protocol that takes 1 hour. And if we wanted fully sampled shells for each of them, it would be 30 hours. So it's completely unfeasible. If we really want to explore the diffusion weighted space, we need to do some sort of undersampling. Its shells are not really realistic. And as an example, if we look here, b-value 6000 and diffusion time 84 milliseconds, we have 60 directions there. The simplest rotation invariant we can build is to take the mean over the measurements, right? The mean over the sphere. And that's what we call S0. It looks like this. It highlights the white matter because it's high diffusion weighting. And if we look here, this rotation invariant that I'm building, it contains only one direction sampled at this b-value and diffusion time combination. But unlike this one is using the neighbors to inform about the directionality and how to compute this invariant. And this also works for different diffusion weightings. And you can also go to higher order rotational invariance. It's not that you need to stick with the isotropic part. Here, if you compute the variance of the signals over the sphere, that's kind of an addition of all the higher order invariants. And here I'm showing S2, which is kind of the main contributor. So you could say that this is roughly spherical variance. And of course, this highlights the white matter. And you can see that it matches really well. Again, this may be a bit misleading in the sense that this one uses 48 directions, and this one is not that it uses one direction. It just has one direction sampled here, but it has 549 that were sampled at different p-values, right? It just, and the method here is exploiting the fact that you have directions somewhere else, and that's typically not the non-trivial part. And yeah, the maps looks really good even at 1 millimeter isotropic resolution in a Prisma scanner. And here I wanted to highlight that we never assumed anything here about the functional form of the kernel. That's why this can work in both gray matter and white matter. So the kernel here could have had exchange, could have had 2, 3, 10 compartments. It could have finite axons. It really doesn't matter. We are just assuming a spherical convolution for this to work. And that's kind of, I think, the nice thing about the framework. So now, I talked a bit about quantitative MRI and like both on the representation side and the modeling part. I showed you some ways to do faster diffusion MRI. I put this because otherwise I would not drink water. This is my first time in the day. Okay, that was worth it. And now I want to talk to you a bit about high resolution, right? Because in the spirit of undersampling, maybe we can keep pushing for that and try to gain also resolution, not only coverage of the experimental space. And the idea is, again, I have people that know way more about this than me here, but in MRI, we measure spatial frequencies, right? We measure the K-space and then we convert that into images. And as we increase the K-space coverage that we're measuring, the frequencies in K-space, the reconstructed image looks with better and better resolution, right? The downside is that, of course, acquiring this amount of data takes more time than this. So we don't want to do that. And that's why there's a whole subfield in MRI that works about doing K-space undersampling, right? Essentially acquiring fewer K-space points and try to get at least similar quality images. And this has shown a tremendous amount of progress for single images. But the point is that here in diffusion, we're never really acquiring single images. Although the current paradigm kind of thinks it does. In current diffusion weighted imaging, essentially, if we're acquiring, say, b-value 1000 along the x direction, we acquire these K-space points. If we acquire along y, we acquire the same K-space points, and we inverse Fourier transform to get the image. If we acquire this direction along c, we acquire the same K-space points, we do the reconstruction, and then we get the image. And we do the same if we change the b-values. And this is extremely inefficient because essentially we're solving the problem of image reconstruction independently for each image. And essentially we're ignoring the redundancy of images. And the bad thing about this is that we don't really care about reconstructing each image. We care about the maps that we're gonna get from the images later. So essentially the actual bar for sampling frequencies for each of these is a bit lower. And the idea is why not keeping the spirit of Q-space undersampling and undersampling both K-space and Q-space. So we can do something like this, where each time we change the diffusion encoding direction, we also change the K-space sampling. Each of them is highly undersampled, but when we combine all of them, we have a fully sampled image. And the idea is that we can do a joint reconstruction, and the glue that is holding everything together is the spherical convolution, right? Because essentially we can see that each direction has a sort of like a coil, similar to what we do in grappa. And I want to show you now, I'm almost done, don't worry. I know you must be super hungry. To talk to you a bit about the future lab research. And I think that the main pillars I'm trying to focus on is, of course, accelerated microstructure imaging. And the first one is study of pathology. I'm focusing on an R00 starting next year to track multiple sclerosis progression and detecting small lesions using this high resolution sequence. But I'm also interested in other pathologies like MTBI, schizophrenia, post-traumatic stress disorder. But also I'm interested in going away from the brain a bit. I have some current projects on muscle microstructure, studying atrophy and sarcopenia on the calf, or also studying myofascial pain in the face. And another thing that I'm really interested in is studying function. Even though I don't have too much experience on functional MRI, I really want to start working on that. And I feel like also it will be something interesting to explore to try to replicate some of the things with diffusion functional MRI. And I think that both of the domains kind of could definitely benefit from either KT or KQT undersampling. So I think that maybe a lot of things of this KQ undersampling that I was telling you from before can be translated. And of course, a lot of work on diffusion from MRI validation is to still be done. I would be more than happy to participate on that. And of course, I'm also interested in going beyond diffusion and work on other on other MRI contrasts, right? I show you some of my work in diffusion relaxometry, but there's no need to stop there, right? Microstructure drives all contrast mechanisms, so we could do diffusion relaxometry susceptibility, for example. And the nice thing about measuring different contrasts is that it gives you extra potential for acceleration, right? Because the microstructure that is generating them is the same. So you can do joint modeling and joint undersampling. And yeah, I think this is full of technical translation and challenges, and I think that that's why I'm kind of interested in it. With this, I wanted to summarize, and yeah, as I told you, if we can do better modeling, this can lead to better undersampling, and hopefully with this we can get faster and more informative scans. Sometimes we learn to do the simple things, but by first doing them a bit harder. I showed you the rotation imbalance of the cumulative expansion, and I showed that this is a complete microstructure fingerprint up to order b squared. And then I think is that it gives us additional sensitivity, white or gray matter. And I show you that with the spherical convolution framework, we can actually exploit redundancy in K and Q space to gain speed and resolution. And I show you some of my plans of accelerated microstructure. This, I want to thank you for your attention. Sorry for keeping you away from pizza. And yeah, that's pretty much it. Thanks.
Wonderful talk. We have actually time for questions.